Cedars-Sinai talking about the orphan conditions. We saw that incredible picture from Dr. Steinberg. I didn't have any triple backflips. That was really a stage winner. That's why he should have ended with that instead of the end here. Talking about mice and urine later. That's not a good way to end. But you know, looking at, for example, with some of the brain tumor patients, we have a drug that is commonly given to brain tumor patients. The goal is to shrivel up blood vessel growth because you want to kill brain tumors. But if they also have moya, you want to encourage blood vessel growth. What do you do? And so we've studied, you know, how that works with kids that have brain tumors and this type of medicine. And you know, we have all these different collaborative groups. We have a working sickle cell group. We just had a meeting at our last National Neurosurgery meeting from 5 different centers, you know, down in DC, in Baltimore, in Michigan. Folks that see a lot of kids with sickle cell disease, where there are large sickle cell populations. And we're trying to put together a collective study section to say, well, how can we as a group learn better about these patients with moya than we could individually? And these are ways of trying to deliver on these collaborations to be smarter about what we're doing collectively. As an example of this, you know, you've seen these moya slides. These are the blood vessel types that are thickened here. And, you know, why does this happen? Well, as Dr. Steinberg has his collaborations with some great folks out in Stanford, we were lucky to have Dr. Sahin here, and you know, we do these cheek swabs on some of our patients, and amongst other things, you know, we found a number of different genes, some of which are similar, some of which, one of which I'm very interested in is TIMP2, which is a gene that regulates the way blood vessels interact with their surrounding environment. And this is particularly exciting for me for my research that I do myself individually. But this is an example of how these collaborations that might otherwise not exist really come to bear by working with folks, you know, in collaborative ways. The big question that I think, and I mentioned to you earlier, is how can we do better? I mean, you hear what these two folks have done with their surgeries over the years. It's astonishing. And, you know, again, the Larry Bird, Michael Jordan, Ted Williams, Joe DiMaggio, whatever the sports analogies you want to have, I can only hope to be as good as surgeons as they are with years of practice in their training. And to improve upon that and to get better than those minuscule, you know, rates of problems, I think there's still room to get better. For everyone in this room that's had a problem or complication, who's had a stroke before they came to surgery, I think our onus is on us as physicians who know this disease to figure out better ways. Can we find out how the disease is present beforehand? Can we treat them better? And so this is one of the things I've tried to pick up in my laboratory and really focus on it in a patient-centric manner. One of the biggest things that I think everyone probably in this room is well aware of is the importance of early detection. You know, if you could find out you had moya before you had a stroke, wouldn't that be great in the sense that you would no longer hopefully have the concern that you'd have to recover from those bad injuries? And you saw Dr. Scott's case of that young girl from Iceland who, you know, had a great response to surgery, but unfortunately was left with deficits before she ever knew she had the disease. And so one thing we've tried to look at is figuring out who might have moya from an early stage. And moya is so rare, is that really practical to do? Hard to tell. But the other thing is prediction. If you be able to, you know, figure out which folks would respond well to surgery or certain kinds of medication and know them better. And so we've talked about this where really the name of the game is early detection. If you can find out you have the disease early, if you know which folks are going to respond best to surgery, that's where you can have a tremendous impact and where I think you can marry what's going on in the lab with what's going on in the operating room. And so is there actually data to support this idea? I think you've heard some of it already. We've looked at our groups here and we said, well, let's look at people that get scanned for other reasons. So kids that have brain tumors, neurofibromatosis kids, where they get scanned because of their NF, not because of their moya. Or kids that have unilateral disease, they have moya on one side, and you're following them along with scans, and you know they might get moya on the other side. And when we look at those groups, we find that their diagnosis when they do develop moya is they have much lower Suzuki grade, so their narrowing isn't as bad, and they have up to a 6-fold lower incidence of strokes. So what that tells us is early detection helps. You can really make a difference if you can find the moya early. This is hard data. This isn't sort of, we wonder if it works. This actually has been shown. So there's a real impetus to get better at finding things early. And again, Dr. Scott already showed it, but this is from our sickle cell group here. This is Dr. Scott's data. That's from our New England Journal little picture here in terms of what the blood vessels can do. And what we show is if you can, you know, treat them with surgery, you can really shut down the amount of strokes they've had. Dr. Steinberg's shown that, Dr. Scott's shown that. Our problem is getting them before they've had those problems. Are there better methods to screen for moyamoya? And so what do we do now? It's the best we have, 2011, you know, here in Philadelphia, at Harvard, at Stanford. Well, the best we really have in many ways are scans. And as good as they are, there's some problems to them. You have to think about getting a scan in the first place. For kids, you have to sedate them. For kids and adults, there's radiation involved. It costs a lot of money. And the question is, can we improve on this? We're not going to get rid of scans, but can we find something to complement them? And so one thing I've been interested in my laboratory is biomarkers. You know, the idea of looking at proteins or chemicals in different fluids of the body, in urine, spinal fluid, and blood, that might be able to tell us what's going on without waiting or having to get a scan, or maybe tell us, maybe, gee, you should get a scan. And so where do you look for these? Well, everyone likes to talk about spinal fluid, but to get spinal fluid, you got to stick a big needle in the back, and that's just flat out not fun, and frankly not practical to do on an easy basis. Blood is easier to collect, but you still have to get needles in people, and there's a lot of background noise. You know, there's a lot of protein in blood, sugar levels go up and down at different times that can bind to some of these proteins. It can make them a little noisy to detect. And that leaves urine, and no one likes to talk about pee. I've been consigned to talking about urine, which is nice, not school at least, which is a plus. But that yuckiness aside, you know what? People pee every day. It's easy to collect. There are no needles involved, and the kidneys filter out a lot of the background noise. So, and I've highlighted it in yellow, which is funny. Probably laughing. But you know, I really think that this is something where there's a lot of practical application, and as a surgeon, I hope to try to be. My wife doesn't think so, but a very practical guy, and this is something that I think is a useful point. What would we look for? So if we look in the urine, what are we looking for in the urine? Well, what we're looking for are a certain number of chemicals. I started and did a sort of postdoctoral fellowship in a laboratory my primary mentor is a woman named Marcia Moses. She's incredibly bright and a very accomplished. She's now the director of the vascular biology program at Children's and full professor at Harvard Medical School. She's just a great scientist. Her mentor was a guy named Judah Folkman, who was a gentleman who since passed away, unfortunately, but really was the founder of this blood vessel growth study. This is called angiogenesis. And they together, sort of, headed by Dr. Folkman and subsequently Dr. Moses, found a number of these proteins and chemicals that are involved in the growth of blood vessels. This little cartoon showing this. They looked first in tumors, because this is something where when tumors grow, they have to elaborate these. Basically, they have to send out these fertilizers to get new blood vessels to grow. And Marcia's real contribution, Dr. Moses' contribution, was to get practical and look for them in the urine. Say, could you get a urine test for prostate cancer, for colon cancer? And so she's looked at this in a number of different diseases, and when I first started at Children's, Dr. Scott, I gave him great credit not only for being a great surgeon, but he said, Ed, why don't you go talk to Marcia about urine? And I kind of, okay, sir, that'd be great. But it really was the start of a wonderful relationship with her in the laboratory. And essentially, this is one of our animal models here. What we're showing, and I've done work in tumors as well, these little bubbles right here are both little tiny tumors, and that black bar is about 1 millimeter. So we like to make things small in neurosurgery. And what's important about that size is tumors that size or smaller can survive just by osmosis. They can just kind of grow, their stuff washes out, their waste, they suck in oxygen, not a problem. But if they want to get bigger than that, if they want to grow, beyond that size, they got to recruit blood vessels. And you can see here, this blood vessel has started to, this tumor has started to recruit a bunch of blood vessels in there. You can see that pipe coming in. And whenever they do that, there's a very sort of replicated, conserved, sort of panel of proteins that come out called the angiogenic switch. That's the, if I'm going to grow, I got to make this stuff. And this is our first panel of molecules. We've now gotten a lot more that we've narrowed down. But when these molecules come, they travel through the bloodstream. They can be detected in the blood, and ultimately the blood goes through the kidneys and end up in the urine. So this is a starting point where we can say when something's growing new blood vessels, in tumors first, and now with moyamoya, where you see those blood vessels and there's a starved brain for oxygen, just like this tumor is starved for oxygen, are those same molecules around. And so we decided what molecules to pick based on some other science done on tumors and other blood vessel problems. We picked this initial panel of 4 molecules. As I mentioned, we have more now, but we started with this. And I asked a very simple question, because I'm kind of a simple surgeon here, but our hypothesis was, if there's a panel of these proteins that can detect different starved conditions, ischemic conditions, tumors outside the central nervous system, And if these same processes of starved tissue, like the brain starved for oxygen and blood vessel growth is occurring in moyamoya, well, then could you use a urine test with these same markers to distinguish between folks that are healthy and those that might have moyamoya? So pretty straightforward question, I think. We've looked at this in tumors, we've published this. We just found out about a month ago that we're starting a national multi-center trial through the PBTC, which is a pediatric brain tumor group, looking at these biomarkers for brain tumors. And with this study, what we're trying to do now is, can we do this in moyamoya? And so what we do, basically, folks come in with moyamoya, we get a collection of their urine. We now have a big bank, actually over 400 samples of different types of moyamoya specimens, and then we analyze them for the various proteins we have there. And so we asked some simple questions. I don't know Dr. Steinberg well enough to say it. Same as a child sitting from the Dr. Scott. That's me. No, we don't know who that is. So the question is, can these molecules be detected in the urine? That's the first question. Yes or no? And what we find is we take healthy people, those are the white bars here, and we take people that have moyamoya, and we just say, are these molecules there or not? And I think you can see even in the nosebleed seats way in the back, there's a big difference between those that have moyamoya in the black and those that don't. It's a good sign that, number one, these molecules are detectable in the urine, which is important. Do they get in the urine at all? And the answer is yes. And two, is there a difference between them? We then ask, this is kind of like the game Clue, is it makes sense that what's going on in the brain, whether it's a tumor or moyamoya, is that stuff actually the stuff that gets out into the urine? And so it's kind of like the game Clue in that you say, well, can you link the chain of evidence? Is Colonel Green in the kitchen with a candlestick? Well, can you find the same tissue specimens from the brain all the way down to the urine? So we first did this in tumors. And what you're looking at here, these are levels of the proteins in the urine and the spinal fluid for a given patient. And then this is that same patient's tumor stained for these same proteins. And so you have different patients working their way across. And what you find is when you look at how the tumors express their proteins, which you can see here, for example, with this patient, they had a lot of this protein. There's a lot of it in the brain tumor. So you see it in the urine, you see it in the tumor. There's very little of it in the urine. There's very little of it in the tumor. With a different kind of tumor, the opposite. You see there's very little of it in the urine, very little of it in the tumor, a whole lot of it in the urine, a whole lot of it in the tumor. So this concordance, it matches, it's sort of got that chain of evidence between the top of what's going on in the brain and the bottom of what's going on in the urine. And we were able to do that with moyamoya as well, where we can find the same markers in the blood vessels from moyamoya patients and carry that out into the urine. So we have that kind of data that we can follow along. One of the other questions people always ask are, how are the statistics? I mean, okay, it looks nice, you can always get maybe one patient that looks really good, but can you really, with numbers, validate this over time? And the important thing here is what we call